Hello and welcome to the lecture on cancer for Bio 110. Here in this slide, what we have are the objectives of this section. Remember, this section only covers part of the material that is covered in chapter 20, so you do not have to read the entire chapter, just the portion that describes cancer. And I'm going to leave these objectives here for you to examine on your own. Now, as we have discussed before, when we have a factor that is triggering a signal through a growth factor receptor, that cell it's going to undergo cell growth. So now the cell growth is going to depend on the extracellular environment. It's going to have limiting, like in the amount of nutrients that are present for the cell to actually grow. You're going to have those extracellular signals that are going to activate the signal transduction pathways to instill cellular growth. And we're going to look again about the issue of mitogens, which are going to be molecules that promote cell proliferation, growth factors that induce growth of the cell, and survival factors that are going to block apoptosis. Now, when we think about the growth factors and mitogens and how those factors are activating signal transduction pathways, we're going to see that they are going to induce synthesis of macromolecules like proteins, and they're going to also prevent protein degradation. Now, when we look at this image, we have here in dots many of the intermediate molecules that are involved in a signaling pathway, but in green circles we have the major components of those signals. And many of the signaling pathways are mutated or affected during cancer. For example, growth factor receptors can turn on receptor tyrosine kinases, and those receptor tyrosine kinases, like the HER2/neu or the growth factor receptor, are going to be affected, and eventually they turn RAS. And those activation of RAS, as you know, can mediate the turning of the cell to growth and therefore induce cancer. We're going to be also be looking at the retinoblastoma inhibitor protein, which is going to be one that controls cell growth. And many of the pathways that are going to induce proliferation are going to do so by inhibiting retinoblastoma and therefore allowing the cell to grow. But if retinoblastoma is inhibited and mutated on its own, it can lead to transformation of cells. Last, we're going to be looking at DNA repair mechanisms and how those are going to activate the molecule p53. As you know, p53 turns on p21, which inhibits cyclin-dependent kinases. That is also going to eventually have its effects on apoptosis. So if the cell cannot repair the DNA, the apoptotic pathway will be triggered. But you can also look at death factors and how those death factors can induce cell death, or the effects of blocking or activating BCL2 to prevent apoptosis. Now, when we think about cancer is the general name for many different diseases that affect many different tissues. And however, those different diseases have properties that are in common. And the first thing that they have in common is that they are uncontrolled cell growth. So when we look at the properties of cancerous cells, we look molecularly at the dysregulation of cell proliferation and differentiation. So the cells are no longer responding to the proliferation signals that the cell gives, or the control of those signals, and therefore they proliferate uncontrollably. They also could be induced for differentiation. Other properties of cancer cells include the prevention of cell death by apoptosis. So activating molecules like BCL2 can lead to the salvages or the rescue, of cells through apoptosis and therefore prevent them from dying, which leads to cancer. Other ones are going to have genome instability, and the genome, when it's unstable, it's going to have a tendency to accumulate mutations. The last thing, it's the issue of metastatic cell growth, the capacity of tumor cells to be able to invade and attack other sites of tissues that are different from the ones in which they generated. Last but not least, we have the issue of angiogenesis. A tumor cannot grow if it doesn't have nutrients and oxygen. So therefore, oftentimes tumors are going to allow the generation of blood vessels that are going to provide them with nutrients as well as oxygen. Now, when we look at the features of cancerous cells, we can think of them as having the capacity to proliferate uncontrollably and the capacity of invasion. So now, self-sufficiently in growth signals, meaning that the cells are not requiring growth signals to grow, therefore they're able to grow in the absence of those signals and doesn't require the system to provide them for them. The cell is going to have insensitivity to anti-growth signals. The control signals that are usually designed to prevent cells from growing are no longer capable of controlling those cells from growing. So in that regard, we know that cancer cells oftentimes bypass many of the cell cycle checkpoints that we discussed earlier. At the same time, we have the capacity of cancer cells to evade programmed cell death, and therefore are able to survive longer even when the system is instructing them to die. Oftentimes, cancer cells are able to have limitless replicative potential, also meaning that the usual time clock, i.e. the telomeres that are going to maintain a cell and tell it how long it can divide, are no longer applicable to cancer cells. They activate the enzyme telomerase and therefore are able to maintain a constant length of their telomeres. Oftentimes, depending on the stage of the cancer, a cancer will develop the capacity to invade a tissue and establish a new tumor, and as you know, that is called metastasis. And when it gets to the new site, it needs to be able to bring new blood vessels to establish a proper tumor, and therefore they can sustain angiogenesis. So, angiogenesis, as I mentioned, is the formation of blood vessels that are going to bring nutrients and oxygen to the tumor. Usually, a cancer cell doesn't stay in the location that it originally started, but is able to migrate to new tissues and form new colonies. So in figure A, what we have is a cancer on an epithelium. In white, we have the epithelial cells. In yellow, you have the basal membrane that is separating the epithelium from the connective tissue in blue. And below, as you can appreciate in the connective tissue, there is a vessel. Now, a tumor cell that starts to grow in an epithelium eventually will be able to break through the basal membrane and invade the connective tissue, and once it invades the connective tissue, it's able to go into the blood vessel. So the cancer cell, once it is able to go into the blood vessel, after that it's able to then exit the blood vessel and invade a new location. So here in the middle, what we have is a liver from a patient that had colon cancer, and what you see inside, it's the secondary tumor metastases that were generated from the original colon cancer cells. The problem is that, as you can see, the liver has lost most of its tissue, and therefore now it's colonized with cancer cells, and therefore it doesn't perform the functions that it needs. In the right figure, I want you to show you the normal tissue of the liver, shown here in the bottom part in this H&E image, and then the very dark purple color cells, which are the tumors generated from the cancer cells. So one of the things that the tumor cell is going to do is to eventually gain the ability to migrate and also invade other tissues. But that is not an ability that all cells have, and it is acquired during the carcinogenic process. Now, when we think about a tumor that is confined to its original site, we term it benign. And one tumor that is able to then exit its original site, it's going to be considered malignant. So here on the left, we have an adenoma. An adenoma is a tumor of a glandular tissue. And as you can see, you have the normal glands, tubules shown here in the cross section, and in one of those you have now the neoplastic granule cells that are starting to grow. But in gray around this tumor, you have this fibrous connective tissue that is encapsulated. That is the basal lamina, keeping it nicely tight and contained. So those benign tumors are able to remain within the basal lamina and therefore do not ever get the capacity to migrate out. and therefore we call them benign. They're easily able to be taken out and contained within it because they have not developed the capacity to break outside of that boundary. The malignant cells, however, on the right, are able now to acquire mutations, or were able to acquire mutations that allow them to exit the original tissue. So they, for example, can destroy the connective tissue around the tumor so that basal lamina is digested away. They're able to invade the connective tissue in gray and eventually migrate through a vein and enter circulation. So in that case, that will be an adenocarcinoma, which is considered malignant. Now, for a cell to become cancerous, it has to undergo several genetic processes, and many of those quote-unquote accidents that can affect the cell are going to affect genes important in cancer. So we call these genes proto-oncogenes, and proto-oncogenes are genes that usually have some kind of role involved in control of cell growth or cell division. When those genes are mutated, they turn into oncogenes, and the oncogenes are the genes that mark many of the cancer cells and are usually shown and affected in those cells. So for a proto-oncogene that is functioning normally to become an oncogene, it can have one of several different events. For example, here in the left, you can see that the proto-oncogene underwent a mutation. So it could have been a deletion, an insertion, it could have been some kind of mutation in the coding sequence that now renders, once the protein gets translated, renders that protein hyperactive even when it's made in normal amounts. For example, if you have a mutation in RAS that prevents it from hydrolyzing GTP, RAS will be completely active all the time, and therefore that RAS mutated protein will continue inducing signals to allow the cell to proliferate. Oftentimes, proto-oncogenes are going to suffer events that are going to replicate them, and therefore increase the number of those genes that were in the genome. So we term that gene amplification. So usually in the chromosome you may have only one copy of the gene, and in this example here in the middle, now that gene has been duplicated and made two copies of it. So now, instead of having the normal amount of protein that the cell requires, you have 3 times as much, and you have now an overproduction of that protein. That also can lead to a carcinogenic phenotype. Another example over here, it's going to be the chromosomal rearrangement. So for example, you can have a gene that usually is not expressed in a cell, but because the chromosomes have undergone rearrangement and an active promoter region has been put next to the gene, you're going to now have an increased amount of that normal protein to be overproduced. So the normal regulation of that gene is no longer working, and you have now this issue of having a new promoter that is constitutively active, making a very large amount of normal protein. On the other hand, you can remove control completely whatsoever and make a gene fusion. So here in the right, by chromosomal rearrangement, you can put a regulatory region that is going to now, here shown in red, have a different function. And when you make this fusion protein, for example, it could be now super hyperactive and therefore lead to the production of cancer. One other example that is not shown here is the disruption of the promoter regions. So if you have a sequence in the promoter that prevents the gene from being transcribed, and that sequence is now modified through mutation, that regulatory region no longer works, and therefore you have active transcription of the normal gene at the times that it's not needed. So that also leads to overproduction of the genes. Here at the bottom, what I have is a website, thelearner.org, that has animations explaining some of the ways in which proto-oncogenes becomes oncogenes. So take a look at that because it's very informative. When we're thinking about gene mutation, we can have 2 different types of gene mutations. One of them is going to be the gain of function. That is going to take a proto-oncogene and enable it for exhibition. On the other hand, we can have a recessive mutation, which we call a loss of function. So let me walk you through this so you understand what I'm talking about. On the upper part of this graph, what we have are showing is a cell with its 2 chromosomes, and in the light blue area, you have the proto-oncogene. Now, that proto-oncogene, for some reason, has been mutated into an oncogene, and now you have one chromosome having the normal proto-oncogene, and the other chromosome having the mutated oncogene. So all of a sudden what you have is the activation of that mutated gene. It doesn't matter that the other copy is working normal. Just because you have now mutated one of them, the oncogene is able to produce and exert its function, activating cell survival, proliferation, or sometimes both. And therefore, you have an excessive amount of cell survival and proliferation leading to cancer. On the other hand, in the bottom part, what you have is a recessive mutation, and that is considered a loss of function. And those usually happens with tumor suppressor genes. So now, in the cell, you have 2 tumor suppressor genes shown in the chromosome, and a mutation, shown here as a red box, is going to inactivate one copy of a tumor suppressor gene. Now, that cell, because it still has a normal tumor suppressor gene, will not turn into a cancer cell. So you require a second mutation that is going to effectively override and destroy the tumor suppression function of those genes. And because of that, the 2 inactivated mutations basically functionally eliminate the product of that tumor suppressor gene, and that is going to now promote cell survival and proliferation, which results in a cancer cell. Now, let's take a look at some of the ways in which tumor suppressor genes can be activated. So for example, here again, we have a cell which has a normal tumor suppressor gene, shown on the right, and a loss of function mutation in the left chromosome, which happens to be the maternal chromosome. So you have a good working tumor suppressor gene on the parental chromosome and a loss of function mutation on the maternal chromosome on the left. So now, on the first example, you have a problem in chromosome segregation, and therefore you have now a cell that lost the parental chromosome, only letting the maternal mutated chromosome behind, and therefore having no suppression on the tumor. In the second example, you have now the maternal chromosome having the loss of function mutation, but you have a deletion of the normal gene containing in the parental mutation, and therefore you lost the capacity of that cell to make the tumor suppressor gene. And that leads again to the formation of a tumor. On the third example, you have now a second loss of function mutation in the parental gene that now inactivates it. So when it's inactive, in both the maternal and paternal chromosomes, the cell no longer is able to suppress the activity of that tumor suppressor gene, and you lead to cancer. And last but not least, on the right, what you have is an effect by a different gene that is going to now silence, by epigenetic changes, the activity of the parental tumor suppressor gene. And therefore, effectively, even though the gene has not been mutated, it has been silenced, and therefore no protein is going to be made, and that leads now to the inability of that cell to suppress the tumor, and therefore cancer emerges. So when we take a look at what causes cancer, we have to think about the fact that cancer is a complex disease, and it's going to have a genetic component, you're going to have an environmental component, and you have effects from pathogens. So now, environmental exposure to ultraviolet light, for example, or mutagens, can damage DNA and affect a cell's capacity to control cell growth. Infection with certain viruses, like the human papilloma virus, hepatitis virus, the Kaposi sarcoma virus, all of this can eventually alter the capacity of the cell to control cell growth, because the virus stem cells are even inserting themselves in a regulatory region, or they are producing proteins that inhibit cell control of proliferation. Last, you can have defective genetics. For example, in children, you can have the inherited of a mutated gene from one parent, and therefore you only require a very slight mutation in the healthy gene that is going to then lead to the onset of mutation like retinoblastoma. Or you can have inherited the potential to have problems with a gene, like in the case of breast cancer. At the end, what we're going to also see is that cancer away from children, it usually leads in the accumulation of multiple errors in its cell function, and therefore the total number of mutations that is accumulated with age can lead to the dysregulation of cell growth, which eventually leads to formation of the tumor. Now, when we take a look at some of the chemical agents that cause cancer, we call them carcinogens. Some of those are going to be chemical, molecules like aflatoxin B or benzopyridine that eventually are going to affect DNA, or you can have the effect of radiation. For example, UV radiation can induce thymine-thymine dimers, and when the DNA is replicated, that thymine-thymine dimer is going to be read as only one base, inducing a 1-point deletion. Or X-rays, which can induce double-stranded DNA damage. You also have the effect of viruses, when viruses oftentimes, as I mentioned, encode proteins that are going to affect normal cell growth and therefore lead to eventual cancer. An example of one of these viruses are the human papilloma viruses, particularly human papilloma virus 16 and human papilloma virus 18. As you know, those viruses cause warts, and in the case of tissue, as you know, a wart is infection and proliferation of the epithelium. So in the case of the skin, you have a HPV virus that is going to infect the epidermis and go down to infect the basal layer of cells. That is going to induce the proliferation of the cell because the virus contains two proteins, one called E6 and the other one E7. And those viral proteins are able to make the cell proliferate. So what you have in the formation of a wart is the tissue that is proliferating and therefore allowing later the release of the human papilloma virus outside to the environment for the next person. Now, eventually, the cells who are now stimulated by the E6 and E7 proteins to proliferate can break down the basal membranes and go down to the dermis and therefore become tumor cells. As you know, when we have talked about apoptosis, we have think about the capacity of X-rays to break down DNA by inducing double-stranded DNA breaks. And the way that the system understands us, it's by having protein kinases that are going to phosphorylate p53. Now, p53 is usually bound to a protein called MDM2, and that protein is targeting p53 for degradation in the proteasome. When DNA damage is detected, kinases that are able to bind to double-stranded DNA will phosphorylate p53, and that will free p53 from binding MDM2. Now, the phosphorylated active p53 can travel and bind to the regulatory region of the gene p21, producing the p21 protein, which is the CDK inhibitor protein. That protein is able now to bind active G1S or CDK or SCDK and inactivating it, therefore putting pauses in the cell cycle. So if the DNA damage is beyond repair, that is going to turn on the activation of pro-apoptotic genes. This is one of the things that also papillomaviruses affect. So as I mentioned to you earlier, the protein E6 and E7 from human papillomavirus are able to inhibit the function of p53, which therefore is unable then to turn on p21, and p21 cannot pause the cell cycle. So by having the proteins from human papillomavirus, the cell is now unable to stop the cell cycle and continues, and that can lead to problems of aneuploidy and DNA damage because the genome has become unstable. Another one of the targets that is affected by these proteins, in particular affected by E7, it's the retinoblastoma gene. The retinoblastoma R gene, shown here by PRB, it's a gene that can inhibit the factor E2F, and E2F is a factor that can stimulate cell cycle control. So traditionally, the normal role of retinoblastoma, as I'm going to show you in a moment, is to inhibit E2F. And when E2F is inhibited, the CDK2 is therefore not active, and you will have a pause in the cell cycle. By inhibiting retinoblastoma, E2F is able then to turn the CDKs to initiate cell cycle division. Now, retinoblastoma, it was a gene discovered in children that has the cancer retinoblastoma. And it is a somatic mutation where the child will inherit one mutated copy from one parent and one wild-type copy from the other parent. Through a somatic mutation event, the wild-type copy now it's mutated, and therefore you get a loss of function of the retinoblastoma gene. And that leads to the cause of a tumor in the retina of these children. Here in the bottom left, you can see a MRI picture showing where the tumor in the left eye of this child is. Usually this will lead to blindness because one of the ways to treat retinoblastoma is to remove the eye. Now, as I mentioned, retinoblastoma, it's a gene that it binds to transcription factors and inhibits their function. It is a proto-oncogene. So its normal function is to bind to regulatory molecules like E2F. So when a cell, for example, is stimulated by a mitogen, and that mitogen is going to initiate a signal transduction cascade that is going to activate G1 CDK or G1S CDK, the cell now it's having a signal to go forward with cell proliferation. So the activated CDKs are able to phosphorylate the retinoblastoma gene, inactivating it. That will release a transcription factor E2F that then can later bind to the activatory regions of a gene, initiate transcription, and those is going to lead to cell proliferation. So that will be the normal circumstance in which a cell monitors the function of retinoblastoma by phosphorylating it and therefore inactivating the protein. In the case of a mutated retinoblastoma gene, that gene is no longer able to bind to the product of that gene, is no longer able to bind to E2F, and therefore E2F is constantly turning cell proliferation. So that could be because the protein is mutated or the gene has been deleted and there is no protein made at all. So again, to summarize this portion, let's take a look at what I had said. So first of all, as I mentioned, cancer is caused by many agents, and some of those are going to be environmental, some of them are going to be hereditary, and some of them are going to be viral. Lifestyle can also affect that. That's an issue of the environment, for example, smoking, and some of the factors in the environment can induce mutagenesis. But cancer is a rare event, because usually cells are controlled by the natural cell cycle checkpoints and the immune system to not proliferate. So the event of cancer, it's a failure in multiple different checkpoints that the body has to control cell growth. And some of the features that cancer cells have will include the cell capacity to grow independent of nutrients and growth factors, the ability of the cell to avoid apoptosis by turning on anti-apoptotic genes, and last, the ability to metastasize and induce angiogenesis in a different site. Now, as I mentioned, many cancer cells have activating mutations in normal cell signaling components like RAS, and a lot of the knowledge that we have learned in cell biology can be used to understand cancer and apply novel medical therapies that hopefully will be able to control the disease in ways better than we currently have. So let me tell you about some of the hereditary cancers that we understand now. So some of those inherited genes are genes that influence cancer. For example, the BRCA1 or the BRCA2 are genes that have been identified to be mutated in women that are prone to have breast and ovarian cancer. And oftentimes, through genetic analysis, women whose family history has shown breast or ovarian cancer will get screened, and if they have then the mutated versions of those genes, oftentimes they choose to have preemptive mastectomies to eventually prevent the onset of breast cancer. Now, another gene that is important in cancer that has been the APC gene, and many patients who have colon cancer has been shown to have this gene mutated. So this table here shows some of the human hereditary diseases and cancers that are associated, for example, with DNA repair defects. So I would like you to take a look at this table. Take, for example, in the last graph, the hereditary breast cancer, the BRCA1 and BRCA2 deficiency. Those women have repair in double-stranded DNA breaks by homologous recombination. That is the usual pathway that is controlled by these genes. They have sensitivity to DNA cross-linking agents and are not able to fix those problems with DNA repair. And therefore, usually, they may develop breast and ovarian cancer. Now, let's take a look at some of the hallmarks of cancer, and one of them is going to be the issue of chromosomal translocation. Here on the left, we have an image of the mitotic chromosomes present on cancer cells. And on the right, what we have, it's an immunofluorescent image with fluorescent markers that are able to usually bind to a chromosome. Shown by the arrow is an example of a chromosome who has gone translocation because it has multiple colors. So the chromosome usually would have been completely green, and now in the middle of the green area you have a region of purple. That means that a region from a different colony that usually will contain those genes have been transferred into this chromosome, shown here on the right. That has now put the genes in a different location and therefore develop into tumor cells. You can also have problems during DNA segregation. So the improper segregation of chromosome during mitosis can lead to cancer. So for example, here you have a cell which is about to undergo mitosis. You have the cells in metaphase where most of the chromosomes are aligned the middle of the mitotic spindle, except the top chromosome, which instead of being attached by both spindles, the chromosome is only attached by one. Now, during anaphase, you have the separation of the chromosomes, and as you can appreciate, the sister chromatids from chromosome in the middle and the bottom part are being separated nicely. But the cell on the right now have gotten an extra chromosome, and the one on the left is now missing a chromosome. So you have, after cytokinesis, two cells which are genetically unstable and prone to mutation because they have aberrant chromosome numbers, either because they have more chromosomes or less. Because of aberrant chromosome numbers, you can have an increase in the number of mutations. So let me give you an example in breast and colon cancer. And what you have here are two proto-oncogene receptors. These two, one of them is the HER2/neu, which is a receptor tyrosine kinase, which is involved in cell growth. That's the one on the left in orange. And on the right, you have the epidermal growth factor receptor, shown in blue. Now, what you have in the left, as you know, receptor tyrosine kinase needs to dimerize in order for them to be activated. Traditionally, in the transmembrane region of the HER2/neu receptor, you have a valine, and that valine is required for prevention of spontaneous dimerization. But when you have a mutation that now changes that valine to a glycine, a glutamine, that receptor is able to dimerize in the absence of ligand. When the receptor dimerizes in the absence of ligand, the tyrosine receptor tyrosine kinase domain is able to begin to work and therefore start the signal cascade in the absence of the ligand. Now, on the right, we have the epidermal growth factor receptor. The external domains, as you're aware, are able to bind ligand. But by having a problem of deletion, this receptor has lost the complete ligand binding domain. And now they're able to associate in the absence of ligand and initiate transduction because they have a constitutively active protein kinase domain at the bottom. So in the case of the HER2 receptor, the mutated version is called the new oncoprotein, and in the case of the epidermal growth factor receptor, the mutated version is called the ERB or the ERB oncoprotein. In both cases, you have ligand-independent receptor oncoproteins that are able to initiate signal transduction in the absence of ligands. When we look at cancer cells, either breast or colon, or any cancer cell in particular, we can see that you have an accumulation of many mutations that are going to find in cancer. So this graph from a paper in Science in 2006 enumerates the number of mutations in genes that are involved in different pathways. So what they have done is that they sequenced many of the breast and colon cancer lines available and decided to determine the fraction of tumors that had mutations in a particular pathway. So for example, many breast and colon cancers have mutation in cellular adhesion and motility genes. Many of them, between 75 and 80%, and sometimes with the colon cancer almost close to 95%, you have mutations in signal transduction pathways. Other highly mutated regions are going to be genes that involve in transcription and regulation. Now, to a lesser extent, you have mutations in genes that control transport, cellular metabolism, intracellular trafficking, RNA metabolism. And some of the mutations are either in other pathways not mentioned here or they are unknown. But the main bulk of the mutations in many of the colon and breast cancer studied were in cells that have cellular adhesion and motility, which will promote metastasis, signal transduction, which is going to promote cell division, and transcriptional regulation, which is going to affect both cellular cell division and motility, as well as signal transduction. In order for a cell to undergo mutations in many of those pathways, they have to accumulate over time. And what we have is the idea that cancer disease symptoms don't appear early normally, but appear with accumulation of time. So in this figure, what we have is the annual incidence of cancer in males per 10 to the 5 males with age. And as you see, for many different cancers, we have prostate, stomach, skin, rectum, pancreas, and esophagus, the incidence, which is the number of new cases per age, increases as age of the individual increases. So usually you have very few cancers that are affecting people between the ages of 20 to 35. Very few of them are going to be skin and stomach, but as the and you can see actually no cancers usually of rectum, pancreas, esophagus, or the prostate. But as time passes and the individual is aging, you have an increase in the incidence of those cancers, basically giving us the idea that cancer is a disease of age. The theory goes that you have to accumulate certain mutations with time, and that accumulation of mutations eventually leads to the cancerous phenotype. So for example, in this image, we have an epithelial tissue. That epithelial tissue has a single layer of epithelial cells shown in gray, a basal lamina shown with a yellow line, and the connective tissue at the bottom shown in gray. Now, this orange cell has a mutation that is going to give that cell an advantage. Now, that cell may proliferate because it has going to have certain selection that is going to allow it to proliferate and become and divide more. So it's lost the control of cell division. But that may not lead yet to a cancer phenotype. That is just undergoing what we call hyperplasia. The cell may be growing bigger, for example. Now, after that, that cell is going to survive and proliferate. You will then have a second round of mutation that is going to increase the advantage of that cell to compete with the cells around it. Now, that cell is going to survive and proliferate, and you can see that the cell is dividing and even causing a small accumulation of cells in that area. That still may not be a cancer cell. Eventually, you can have subsequent rounds of mutation, let's say a third, fourth, maybe fifth round of mutation. That is going to provide that cell with multiple advantages in cell division and survival, and maybe even get the capacity to invade and proliferate. So when that cell, now shown here in red, begin to divide, they're able then to probably grow outside of their epithelial layer, past the basal membrane, invade the connective tissue, and now you have the formation of a tumor which is malignant, and eventually with further mutations those cells will be able to escape the original site of the tumor and migrate to other areas such as the bone and the liver. Here in this slide I have an image of colorectal cancers which form polyps in the colon. So here in this picture in the upper left, what you see are the tissue in the colorectal area. So this is the colon, and what you see are many tiny polyps and a very large polyp that has formed. And this looks, I mean, it's sort of, a polyp always reminds me of a broccoli stem because it's going to have a stem and it's going to have a crown. Now, when you take and slide this polyp and do histology, what you see is that you have at the bottom here in the left, you have the normal epithelial layer of the column, but then you have a growth multiplication of that area. So the polyp has able then to break through the epithelial layer and begin to grow outside of it. So now, many normal epitheliums in color are going to have the lack of activity of the tumor suppressor gene APC. So APC is lost, they have excessive epithelial proliferation, and that is going to now increase the activation and proliferation of the gene RAS. So now RAS is active. As you know, RAS is a monomeric G protein, and that is going to now allow signal transductions of the cell. You can form a small tumor. You can now have multiple mutations that are going to eventually lead to the loss of other tumor suppressor genes, and that is going to make the tumor bigger. Eventually, you have a mutation in P53, and P53 in many colon cancers is mutated. That is going to prevent the cell from being able to stop in the cell cycle. That cell now becomes invasive. It is able to form the polyp, and cells from the polyp are able to become metastatic, be able to exit the original polyp, go in circulation, and land in a different tissue like the liver, causing a metastasis to occur. So now, when we take a look at cancer, one of the first things that it's important is to have prevention. So stop smoking, healthy lifestyle, sunblock to prevent skin damage by UV light, but also you have to be tested often because the cancer could be treated successfully if detected earlier. Now, traditional cancer treatments are usually not very specific for a particular cancer. For example, we use radiation. Radiation can kill cancer cells by inducing double-stranded DNA breaks and also affect other rapidly dividing cells, which are not cancerous. Chemotherapy. Most of the mechanisms for chemotherapy are still unknown. We just know that they work, but oftentimes they affect rapidly divided cells. So these both types of therapies can affect normal function. Locally, let me just give you again an example of the HER2/neu gene. The HER2/neu gene has high elevated levels in the serum. So you can actually detect if a person has increased elevation of the new protein on the serum, and that helps you determine the stages of the breast cancer in that patient. Cells which are breast cancer usually express a larger amount of the HER2 protein on the surface and therefore are able to give more signals for cell division. A normal breast tissue cell expresses low level of the HER protein on its surface. So here's when we have a medication, a biological medication called Herceptin. Herceptin is a monoclonal antibody designed exclusively to bind to HER2, the receptor, on cells that express it. So, because of the difference in expression between the normal and cancerous cell, the antibody against the HER2 protein, the Herceptin, is able to bind to the cell and it will target that cell for destruction by the immune system. Rituximab is another similar molecule that is used in the treatment of lymphoma, B-cell lymphomas that express the molecule CD20 on their surface. So rituximab is able to bind to those proteins in the surface of the lymphoma cell and therefore target them for destruction by the immune system. Other chemotherapeutic agents, for example, can block mechanisms like phosphorylation by kinases. So for example, what we have here is a putative kinase. So that's an oncogenic kinase that is active, and it is being controlled by another kinase that is able to put a phosphate into the substrate protein, therefore activating that molecule. Since that protein is able to phosphorylate that substrate protein, protein is going to lead to proliferation and survival and lead to leukemia. In the case of chronic myeloid leukemia, or CML, this is one of the reasons of the MYC protein that is able to phosphorylate and induce cell proliferation. Now, Gleevec, it is a compound that is able to bind to the oncogenic kinase, preventing it from phosphorylating its target protein. When the protein is phosphorylated, there is not going to be a signal and the leukemia cell will not happen. So with that, I have finished the lecture on cancer. Thank you again for your patience waiting for the lecture. And again, thank you for a fantastic semester. I'll be looking forward to see you Monday in class and Wednesday. Bye-bye.